Introduction. Plant tissues play a vital role in the growth, development, and overall functioning of plants. In this exploration, we uncover the characteristics and functions of three important types of plant tissues: Parenchyma, Collenchyma, and Sclerenchyma. Understanding these tissues is essential for comprehending the structural integrity and physiological processes of plants. Parenchyma tissue. Parenchyma is the most common and simplest type of plant tissue. It consists of relatively unspecialized cells with thin cell walls and large vacuoles. Parenchyma cells are typically alive at maturity and can divide to regenerate damaged tissues. They are responsible for various functions, including photosynthesis, storage of nutrients, and gas exchange. In leaves, parenchyma cells form the mesophyll tissue, where photosynthesis occurs. In stems and roots, parenchyma cells serve as storage sites for starch and water. Collenchyma tissue. Collenchyma tissue provides mechanical support and flexibility to growing plant parts. The cells of this tissue contain thickened primary cell walls that are unevenly thickened, particularly in the corners. They are elongated, living cells that often form strands or cylinders beneath the epidermis of stems and leaves. Collenchyma tissue gives young stems and petioles the ability to bend without breaking, allowing plants to adapt to their environment. Its flexible nature aids in primary growth and helps maintain the overall shape of the plant. Sclerenchyma tissue. Sclerenchyma tissue provides strength and support to mature plant parts. Unlike parenchyma and collenchyma, sclerenchyma cells have thick, lignified secondary cell walls that provide rigidity. These cells are usually dead at maturity and do not have a protoplast. Sclerenchyma cells are categorized into fibers and sclereids. Fiber cells are long, slender, and present in bundles, providing mechanical strength to stems, leaves, and vascular tissues. Sclereids, on the other hand, are shorter and irregularly shaped cells found throughout plant tissues, contributing to hardness and protection. Examples of sclereids include the gritty texture of pear fruit and the hard shells of nuts. Structural differences. Parenchyma cells have thin primary cell walls and lack secondary cell walls. Collenchyma cells have primary cell walls that are unevenly thickened. Sclerenchyma cells have heavily thickened secondary cell walls, often with lignin, providing rigidity and support. Functional differences. Parenchyma cells are involved in photosynthesis, storage, and gas exchange. Collenchyma cells provide mechanical support and flexibility to young plant parts. Sclerenchyma cells provide additional strength and support to mature plant tissues. Conclusion. Parenchyma, Collenchyma, and Sclerenchyma are three distinct types of plant tissues, each with unique characteristics and functions. Parenchyma cells are versatile, involved in various plant processes such as photosynthesis and storage. Collenchyma cells offer flexibility and support during growth. Sclerenchyma cells provide rigidity and strength to mature plant parts. Together, these tissues contribute to the structural integrity and physiological processes of plants. Understanding the roles and properties of these tissues enhances our knowledge of plant biology and the complexities of plant life.